Hey, what's up, everybody? We're back. Just doing a part two to what we were talking about originally, dealing with cells. And I kind of want to talk about the organelles inside of a cell. And if you're thinking, what the heck is an organelle? Well, you can kind of take from the name organelle that it must be talking about some little type of organ structures, which you're kind of, you know, safe in assuming so. Cells have these small little things called organelles, and they're little structures that function as the cellular organs. But these organelles function more almost like our organ systems do. Like you have a nucleus that kind of have similar functions to a central nervous system in us. You've got the mitochondria inside of cells, which their sole purpose in life really, well, not sole purpose. Some people, some cellular biologists may argue with me on that, but mitochondria, one of their primary jobs is to produce ATP, energies for the cells to be able to use. So it's almost kind of like, what happens with the digestive system when it processes things to a smaller molecular level so that your cells can manipulate it to be able to use it. And you've just got so many different organelles in there doing their job inside of a cell to make that cell function. So I want to pull up these notes real quick, and I think I'm going to kind of find myself on a bit of a time crunch here due to the fact that my laptop has some limited juice at the moment. If things freeze, just remember it's not you, it's me. I'm just loading some notes from my Google Drive, and just a Google Doc from a Google Drive. And once I get this up and loaded and running, then you'll see me again, and everything should be fluid. Let's skip well ahead. We want to actually go to slide 44, and I want to pull up some things there. And we're not going to take too much time on this. No, really, seriously, if you want to get deeper than this, feel free, go right ahead. Nothing's stopping you. If you got some more questions, just let me know. What I want to do is I just want to do a brief overview of these organelles, because that's the cool thing about anatomy and physiology. You never have to get extremely deep about some of these things due to the fact that you have opportunities to get deeper about certain topics as you encounter them. For example, the mitochondria, we're only going to talk about the mitochondria about this much on this video due to the fact that when we get to muscle physiology, we start talking about muscle fibers and aerobic and anaerobic metabolism, we can really get a lot deeper then. And when you start getting into the digestive system and metabolics and everything, you can get a lot deeper into the mitochondria then. We can start talking about the electron transport system, the ETS. We can start really getting into what happens to fatty acids. We can talk about the Krebs cycle, aka the TCA cycle. We can get into all of that and really expand on that then. But that's then. This now. We just want to keep this very simplex. We want you to be able to see one of these organelles and have a general idea of what's going on. So the first organelle that we have up here is known to us as the ER. We nickname it the ER because nobody really honestly feels like saying endoplasmic reticulum all day long. And if you look at this, we'll just enlarge this just a bit. You'll notice that the endoplasmic reticulum in this image is in two different places. The endoplasmic reticulum is wrapped around the nucleus, and then you'll notice that it's just outside of the endoplasmic reticulum that's wrapped around the nucleus. And you'll notice that there are two different types of endoplasmic reticulum. There's smooth and then there's rough. Really, to be honest with you, the major difference between smooth and rough ER is the appearance. That's why they're called what they're called. Smooth ER looks smooth, so that's why it's called smooth ER. Rough ER looks rough. That's why it's called rough ER. And that rough bumpy thing that's on the of rough ER happens to be ribosomes. And the job of ribosomes is actually to produce proteins. They make specific molecules, and we'll talk about them in just a moment. If you look at these functions of the endoplasmic reticulum, some of it has to do with whether or not it's smooth or rough, and that's just the first one. But typically, endoplasmic reticulum, you can it if it's rough ER, it helps to synthesize or create proteins, which it makes sense because ribosomes create proteins. So if it's studded with ribosomes, then it probably makes proteins because it's covered with the little things that make proteins. Whereas smooth ER is not going to be responsible for making proteins because it doesn't have any ribosomes. Smooth ER is really where you make a lot of carbohydrates, um, and you actually synthesize lipids. So it's a pretty good place for putting together triglycerides and things like that. It can transport substances. Think of your endoplasmic reticulum inside of a cell as a underground subway station. It has subway tunnels that run substances around. So sometimes there's substances that you don't want floating around in the cytosol, but you do want to transport it around the cell. You'll use the endoplasmic reticulum to do that. You can pack and store new substances inside the cell at the ER. You can detoxify drugs and alcohol and poisons. You'll see this actively taking place in the liver. And segments of the ER are pinched off to form transport vesicles and peroxisomes. So you'll see segments of endoplasmic reticulum used for different things. As a matter of fact, if you ever study neurons and you find about these things called Nissl bodies should be little pieces of endoplasmic reticulum that's been pinched off, and they give these neurons this grayish appearance when you look at them under a microscope. So endoplasmic reticulum, that's it in a nutshell. You won't see endoplasmic reticulum moving around anywhere. It's more of a stationary substance. And what I mean by moving around, I mean like wiggling around going somewhere, because the ER is not alive. But it is a mechanism that functions a lot more like a organ system than just a organ. We have our next organelle, which happens to be the Golgi Apparatus. Let me tell you this right now, okay? The Golgi Apparatus has so many names, it's flipping ridiculous. You've got the Golgi Apparatus, you've got the Golgi body, you got the Golgi complex. I've heard so many different names, and I've taught from so many different textbooks. I kid you not. All of my teachers in college used a different name. Each one of them used a different name. The people I teach with, they use different names. I mean, there's so many different people using so many different names. It's ridiculous. I could care less which name anybody uses, just as long as they're talking about the same thing. Personally, for me, I refer to the Golgi apparatus slash body slash complex slash. I refer to Golgi as the UPS center of the cell. And I call it the UPS center because everything that's a function of the Golgi body apparatus complex thing is something that you would see at the UPS center. You pack, you sort, you transport. You know, I mean, that's everything that the Golgi body apparatus complex does. Now, if you're looking around, and some of you are probably looking at the video and you're like, I don't see all those words. No, remember what I said. You can find that the Golgi structure is named different things based off of who wrote the textbook and who's teaching the class. So everybody uses different words. But my main point to all of this is that this structure acts just like a UPS place. We send compounds there so that it can be wrapped up, packaged, and sorted, and then shipped to other areas around the cell as well as outside of the cell. Because there's substances that these organelles are creating all the time that you don't want to have in contact with the rest of the cell. And so you package it in a way so that it does not come to physical contact with the rest of the cell, and that's what the Golgi apparatus can do. We have the lysosome, which the lysosome is a funny little structure to me because the lysosome has a nickname, and the lysosome's nickname is suicide packet. And the reason why it's called the suicide packet is because it's this organelle that's chock full of enzymes, and these digestive enzymes are created to basically break down whatever it comes into contact with. So, for the average cell that has a lysosome, generally it's because they engulf some type of substance that they need to break down and utilize. They expose it to the enzymes of the lysosome, and then it breaks it down. Some cells in your body are trash collectors. They collect trash and debris, and then they expose them to the lysosomes, and the lysosomes break these things down like a garbage disposal so that you could then get rid of these waste substances. And you got some cells that are like the cells in your immune system who have lysosomes, and then they actually use phagocytosis. Remember that term? Phagocytosis is a type of endocytosis, a type of active transport. The cell actually reaches out around and engulfs the pathogen, pulls the pathogen in, and then subjects them to these enzymes that you find in the lysosome, and then it uses it to actually destroy that actual pathogen. So it may find a bacterium and then engulf that sucker and expose them to the lysosomes, and that's all, folks. That thing is dead. Mitochondria, we kind of talked a little bit about mitochondria not too long ago. Mitochondria, they engage in aerobic cellular respiration, also known as aerobic metabolism. Anytime you're talking about aerobic metabolism or aerobic cellular respiration, that is a process that requires oxygen. Generally, we stick oxygen and fatty acids in there and do our do, and we want coming up with all this ATP, which is magical and all that. But the bottom line is that you're not going to get any energy out of this process unless oxygen is allowed to enter into the play, and it must enter into the mitochondria. The less oxygen you have, the more likely things like pyruvate, aka pyruvic acid, is not going to go in and be converted to acetyl-CoA and enter into the mitochondria to make ATP. Instead, what you're going to wind up with is you're going to wind up with that pyruvate sitting there outside the mitochondria looking like he got stood up on his date. And pyruvate's going to get really angry because he got stood up, and he's going to go into a Kanye West tirade and then convert into lactic acid, which is not going to be good for that cell. As a matter of fact, that lactic acid is going to get pumped out of the cell, and hopefully, if all things go well, the lactic acid will get transported to the liver and undergo reverse glycolysis and turn that lactic acid back into sugar, and we try this whole little game again. And hopefully he won't turn into a bitter lactic acid Kanye West molecule. But if all things go well, generally, you pull fatty acids and things of the sort into the mitochondria, and then the mitochondria winds up making lots of ATP. Now, something that's not on this slide that you also probably want to know about mitochondria is the fact that mitochondria also has DNA in it, because we're going to talk about the nucleus. I think the nucleus is like the last thing on my list to talk about. And what oftentimes happens is we talk about the nucleus and we talk about nuclear DNA. And I remember I was in a class, I was teaching this class, and this guy, I talked about, the DNA is in the nucleus and blah, blah, blah. And he said, well, almost all the DNA. And I said, yeah, almost all the DNA. I said, but the DNA that we're talking about today is in the nucleus. And that dude was just frustrated, man. Every time I said something about every time I said something about DNA, that guy got frustrated with me and the reason why he got so frustrated with me was because he wanted me to acknowledge the fact that you do have DNA in your mitochondria. It's not nuclear DNA. This is mitochondrial DNA. And if you've ever watched the Human Family Tree, which is a excellent documentary with the National Geographic. It's not boring. Okay, I know you're probably looking at me saying, well, you're a geek and you like that nerdy stuff. No, really, seriously. Non-geek people really love this documentary because it's not done on a geek level. It's done on an everyday level. And it shows how we can use your mitochondrial DNA to trace your lineage back all the way to figure out where on the planet your people actually came from. So, there's a lot of people out there who are thinking that they came from one place, and really that's not where they're from. And to show that we have a lot more in common with one another than we all believe. But we get it from the mitochondrial DNA. So there is DNA in your mitochondria. Let's see. I think the next thing that I wanted to talk about was ribosomes. And I think that is correct. Yes, it is. Ribosomes, this isn't the slide that I wanted to go to, but I'm going to pause here for a minute. Ribosomes, they are broken into two units, all right? There's a large subunit and a small subunit, and what winds up happening is you make the units in something called the nucleolus. Now, the nucleolus itself is inside the nucleus. So, okay, so you got the cell, right? And so you got the cell, and then inside the cell, you have a nucleus. And then inside the nucleus, there's a nucleolus. And the nucleolus is charged with producing ribosomes. And so if you're trying to envision this without looking at the image, just kind of envision you have a brain, and your brain is like your nucleus. And inside your brain, you have this little structure inside your brain that's responsible for making ribosomes. The same thing happens in a cell, because the nucleus is like the cell brain, and inside the cell brain, the nucleus, there's a nucleolus, and it makes ribosomes and the units, the subunits to make a ribosome. And then the subunits, they get together and they wind up reading things like RNA, and RNA acts as a template or a recipe to create proteins. Now, what kind of proteins? Lots of different types of proteins. I mean, everything from making the pigment for hair color to making enzymes so you can digest a bacon cheeseburger to making all types of different things, to making antibodies. Yeah, I mean, if it's a protein, you probably make it at a ribosome. And ribosomes are extremely important to know about. Ribosomes can be fixed or they can be free. If they're fixed, then they're attached to the endoplasmic reticulum membrane. But if they're fixed, but if they're free, then they're just floating around. And we can enlarge this a little bit so you can see the ribosomes. We'll enlarge here just a bit, and if you take a look, here's the cell, here's the overall cell, there's the nucleus, which is the brain of the cell, and then inside the nucleus there's a centerpiece, kind of like, you know, how many licks does it take to get to the center of a Tootsie Roll Tootsie Roll Pop? So here's the nucleus, and then inside there's the nucleolus, and at the nucleolus that's where you make ribosomes, and the ribosomes are all these little speckled dots, and then you'll notice that they're bound, they're attached to the endoplasmic reticulum, but then you've got several that are just kind of floating around in the cytoplasm, and these are known as free ribosomes. And the free ribosomes, they point them out here inside the cell. These little dots that you see, these are free ribosomes. And what winds up happening is you have what's called a large unit and a small subunit, and the RNA is actually fed. What happens is the large unit and the small unit combine and form a functional ribosome. And what happens is you actually feed the RNA through here like you feed tape or paper through a printer. You feed that RNA right through there, and as the RNA runs through there, the ribosome reads it and then takes amino acids and attaches them together based off of what it just read. It literally reads the RNA like it's a recipe, and it attaches everything together exactly the way it reads it. So you might be making structural proteins. I mean, this cell may be making antibodies. This cell very well may be making antibodies that you are developing because you got the flu shot, so that you can have a stronger immunity against the flu virus. I mean, that's what it could possibly be doing. And I know everybody's got their pros and their cons on the flu shot. I got my pros and cons about the flu shot. So, you know, I'm not trying to start a fight, but But even if you don't get the shot, if I'm standing in a classroom and somebody in that classroom is sick and they've got the flu, I'm exposed to that virus because it's airborne. And so now my body is going to have to make antibodies against that particular pathogen. And what's cool is that, as I'm making proteins, you make proteins for so many different things, not just antibodies, but you make proteins to make hemoglobin for your red blood cells. So ribosomes are extremely important. If you don't have ribosomes, you're going to be in a lot of trouble. Let's see, who else did I have to talk about? I think I wanted to talk about, yes, cytoskeleton. Just wanted to say something really quick about cytoskeleton. The cytoskeleton, let's get right here. I skipped something. Yeah, I sure did. I skipped something. I skipped proteasomes. I'm sorry. Proteasomes, these are organelles that really have two functions, and you're not going to see a lot about proteasomes when you're doing anatomy and physiology. You usually see them more in cell biology, but not in your general anatomy and physiology, because here are the two functions of a proteasome. Proteasomes deal with protein digestion, and you probably picked that up from the name proteasome. Like when you talk about proteases, a protease is an enzyme that breaks down protein. So protein digestion happens because of proteasomes. They degrade proteins that are damaged, incorrectly folded, or those that are no longer needed. So basically what proteasomes do is they take care of old, busted, disgusted proteins. So proteins that you can't use anymore, proteins that are broken, proteins that aren't functional, we actually break those down using proteasomes. And so you take this old, broken down protein, been twisted the wrong way, you run it through the proteasome, and the proteasome takes that protein and snaps that protein apart into individual amino acid and peptide fragments. Now, this is the cool thing. This is what makes it so cool. Amino acids are like Lego blocks where you can just attach them. So you might make the Millennial Falcon out of your you might make the Millennial Falcon out of your Lego blocks. I may turn around and take your same Lego blocks, take that whole Millennial Falcon apart, take those same Lego blocks and make New Jersey. A map of New Jersey, maybe. All right. Same Lego blocks, two totally different things. Okay. Um, you know, um, so I've made New Jersey, I've made the state New Jersey out of the same Lego blocks that you use to make the Millennial Falcon. It's the same Lego blocks, two totally different structures. That's the point of a proteasome. A proteasome takes that old, busted, disgusted protein that you couldn't use otherwise, and then turns it into something that we could use. Okay, my time is running out, and my laptop is really low on juice and don't have my power cord, so we're gonna have to really wrap this thing up. Um, I know we wanted to talk about, there was something that I already mentioned in one of the previous videos, and it was talking about cilia and flagella. And I was talking about the fact that composite image of a cell that's on there earlier in the other video, that composite image of a cell is not a real image at all. You're not going to see a cell with cilia and flagella. That's kind of freaky. So you have cells with either one or the other. Generally, when you see a cell with cilia on it, the first question that you want to ask is this cell mobile, or is this cell fixed? Is it free or is it fixed? Because if it's free, then it uses the cilia to move itself. If it's fixed, like it's connected to a wall somewhere, then it uses that cilia to move other substances across its surface. So the example that they give you are the pseudostratified ciliated columnar epithelial cells in your trachea and along the lower respiratory tract. You want me to say that again? I can't. Pseudostratified ciliated columnar epithelium. If you haven't read anything about tissues and you haven't watched any of my tissue videos, you need to definitely go back and watch some of my tissue videos so you know what I'm talking about. But those cells in your lower respiratory tract have cilia, and what the cilia do is they do this. Okay, they brush and they move the mucus upward. So when you have a lower respiratory tract infection, you have a cold, what they'll do is those cilia beat and they move the phlegm, the mucus, up out of your chest, and they move the pathogens out of your chest, and it moves it up into your throat so that you can spit the sputum out, get rid of it. That's what cilia do. Flagella, the only example in humans that we'll ever see with flagella on it is sperm cell. You don't ever inside of you that has a flagellum on it. That's not a sperm cell. That's not a sperm cell if it's something else and it has a flagellum on it. The only other thing, there's two last things to talk about, and I'm really running low on laptop juice. I mean, the battery sign on my laptop is literally close to nil. But you have microvilli, which are very important. Never forget microvilli. When you get to the digestive system, microvilli are going to be very important to understand, because microvilli look like these finger-like projections on the surface of the cell, and their job is to increase the surface area of a cell so that you can maximize absorption. That is the job of microvilli. You want to maximize the surface area so that you can absorb as much as you can. And the perfect location to find these things are on cells in your small intestine, because your small intestine is responsible for 90% of the food digestion that's going to take place. 90% of the food that you eat is digested and absorbed in the small intestine, and so you need numerous microvilli to be able to absorb all of those nutrients once you've digested them. As a matter of fact, you can tell the different areas of your small intestine from one another based on the amount of microvilli that's lining the walls. And we'll talk about that more when we get to the digestive system later on this year. And of course, the last but not least, is the nucleus. And the nucleus is kind of boring to talk about unless you're going to talk about DNA, but we're going to talk about that at a later date. The nucleus is pretty much the brainchild of the cell. The nucleus is where you store a lot of the genetic material inside of a cell, so that your cell actually knows how to build you and build different things. So when you see the nucleus looks like a little Death Star. Or if you took astronomy or you took geography, you've seen a picture of the Earth, and when they talk about the mantle and the crust and all that, and the core of the Earth, and inside here you can see that the nucleus looks like a little planet, and inside at its core you have a nucleolus, which sole purpose in life is just to make ribosomes, and the nucleus is wrapped up in a nuclear envelope, little things called nuclear pores, which these pores are just big enough to let the newly created ribosomes out, as well as copies, or we should say RNA copies, of your genetic material, so that your cell can then use ribosomes to read that genetic material and then create magnificent things. So that's all I got for you right now. We'll be talking to you later, possibly soon. And you got any questions, post them. Let us know something. Talk to you later. Peace.